Hello everyone, so today I'll be talking about pharmacogenetics.
Pharmacogenetics and pharmacogenomics are both study of genetic influence on drug responses in individuals. The only difference between them is that the term pharmacogenetics refers to a specific gene or group of genes' response to certain drugs, whereas pharmacogenomics refers to the collection of all genes that causes drug reactions. This is a list of what I'm presenting. My personal reason for researching on pharmacogenetics would be that because I would like to understand how genes can influence drug responses. Why do people want it? It improves drug safety and increases drug efficacy and reduces toxicity. It reduces adverse drug reactions and side effects. It identifies optimal dosing, and pharmacogenetics could cure several diseases at the same time as a personalized drug. Pharmacogenomics is more common in the modern days because by personalizing drugs can reduce adverse side effects and reach the level of optimal dosing. DNA plays a large genetic influence on drug response. While medication is given to individuals, the drug may be harmful when there are slow distribution and breakdown by enzymes because it takes a lot of time and energy to be eliminated and causing the drug to react with unwanted regions in our body. Whereas if drugs have quick distribution and breakdown, then the patient can only reach a short efficacy and requires higher dose for the treatment. Best drugs have high efficacy and low toxicity. Doctors use genetic testing with DNA sequencing to see which kinds of drugs best matches your DNA because everyone has a slight difference in DNA like SNPs, which is known as single nucleotide polymorphism. Right drugs or right patients at right dose. Each medication has variety responses on the patient, and if the drugs don't match, it could eventually lead to death because the adverse side effects can interact with the heart problems. A genetic testing on human chromosomes can allow doctors to prescribe the right dose and improve drug safety. Doctors can now predict a drug response on a patient by knowing his or her DNA sequences. This could offer an alternative treatment. Currently, there are personalized drugs on heart disease and chemotherapeutic treatments to reduce the adverse drug reactions. Now, genetic testing can predict how an individual responds to 30 available drugs and for a doctor to prescribe their best dose. There's also the fact that professionals have to expand their knowledge and clinical practices before prescribing individualized medicines.
If genes that code for receptors and enzymes are mutated, then the target interaction, breakdown, and distribution, which are all factors of drug responses, will be affected. So in pharmacogenetics, the genetic testing will see which drugs best suit the individual, and the doctors will prescribe the right drug for the best effect. An example of pharmacogenetics that allows for altered treatment would be the mutation of potassium ATP channel. And this kind of channel is found in the pancreas, located on the beta cells. And the beta cell have three kind of channels. One is the calcium channel and another is the glucose channel. And this one is a potassium ATP channel. It consists of 8 subunits and the exterior 4 subunits is produced through ABCC8G. And interior, the white part, is produced by KCNJ11G. And this allows the potassium ions to move in and move through, and it needs ATP as an energy to open and close the channel. So how this channel works is the glucose enters the GLU2 channel and then moves inside the cell, and then the enzyme metabolizes the glucose and make it into ATP, which increases the ATP concentration inside the cell. And normally, the ATP will bind to the exterior of the subunits of the potassium ATP channel, and that's the channel to be remain closed. And therefore, the potassium ions here wouldn't be able to move out the channel, which on the other side will let the calcium ions come in, and the insulin will be secreted through the cell and moves out. But if the KCNJ11 gene is mutated, and the potassium ion will move out, and because the channel will remain open, therefore the secretion of insulin will decrease. But there is a new kind of drug that is invented from the pharmacogenetics, which is called sulfonylurea drugs. And this will enable the tablets to bind to the potassium ATP channel and let it remain open. Therefore, the secretion of these insulin will be normal, and this results in the normal secretion of insulin. The closure of the potassium ATP channels in response to increased glucose triggers the release of insulin out of beta cells and into the bloodstream, which helps control blood sugar levels. In this kind of pharmacogenetics, it provides an altered treatment for the patients, so the patient wouldn't need everyday insulin injection. Instead, by eating the tablets, they can increase their quality of lifestyle.
Genetic testing is used as a basis of experimental drugs. It determines clearance rate, distribution, bioavailability, and half-life, and it is used to figure out the gene's influence on pharmacokinetics, which are metabolism, and pharmacodynamics, which are drug effects. An example of individualized drug now would be rifampicin. It allows slow metabolism and greater drug exposure, but it has a warning of potential effect. Food and Drug Association approved drugs that apply to several specific SNPs. Pharmacogenetics are especially used in chemotherapeutic for treating cancer. Significance in the future requires improved detection of a change in a common clinical outcome. We want to detect uncommon side effects or therapeutic response. Also, to determine uncommon multilocus genotypes, linkage and structure and frequencies in the polymorphic background across racial and ethnic groups. Genetic marker of drug response must also be replicated in diverse populations if it is to be widely clinically employed. That is why some drugs can be used in crossing regions, because the inheritance in racial and ethnic groups would affect the reactions. There are also a few current clinical challenges. It requires increase of national and society guidelines for common conditions. Medical professionals and pharmacists will need further education and support in interpreting these risk estimates. And it also requires great costs for each personalized genetic testing. This is the data of number of deaths caused by ADRs. There are 2.2 million of people that are influenced by severe adverse drug events per year, and it is the fourth leading cause of death in the US. 100,000 deaths per year by properly prescribed drugs and 80,000 deaths per year by improperly prescribed drugs. We can see that adverse drug reaction by prescribed drugs is rising. The promise of pharmacogenetics is a personalized dose to optimize efficacy and safety. Without genetics, advertised dose can fit all people. I believe the data on the previous two slides can be a reliable source, because many drugs can cause severe side effects that could lead to death if the drug doesn't correctly match the patient's gene. For example, co-dosage of proper drugs could cause severe interaction with mutated genes like drugs of blood pressure and dizziness. This could result in a rapid rising hypertension, eventually causing heart attack. The advantages for pharmacogenetics would be lowering the death rate of prescribed drugs, and improves drug safety by personalizing drugs that benefits and applies to your own gene the best. It reduces great amount of side effects, also can identify drug response and use alternative treatment. Some disadvantages would be the ethics issues. When large amount of experiment is needed, the increase in human treatment in animals will be rising too. The poor people cannot reach this kind of top care. Also, there's complexity to find steps that direct influence drug responses. Bacteria and viruses will evolve and become more resistant to the drug, and therefore, the drug will no longer work for your body. In conclusion, DNA sequencing can be used to examine SNPs that interact with drug responses by using a gel electrophoresis. And using pharmacogenetics,
Humans can maintain homeostasis and make sure the body works at its best. Pharmacogenetics allow people to fit to their individualized drugs in order to reduce side effects.
This is my bibliography.
Thanks for watching.